Each one of your cells is enclosed by a membrane that separates it from the rest of your body or from the outside environment. Within each one of your cells are specialized structures called organelles that allow each one of your cells to function. Remember, if your cells stop functioning, you stop functioning. The plasma membrane surrounds each cell and isolates the cell's contents from the external environment, whether that be within the confines of a multicellular organism or from the outside environment. The plasma membrane is extremely thin. How thin? Well, it's estimated that you could take 10,000 plasma membranes and stack them on top of one another, and they would still barely equal the thickness of a textbook page. Functions of the plasma membrane include to control which substances enter and exit the cell, as well as to transfer chemical messages from the cell's exterior to the cell's interior. An example of chemical messages would be hormones. The plasma membrane is composed of a phospholipid bilayer that is highly fluid without breaking. Bi means two, so there are two layers of phospholipids. Fluid means that the plasma membrane is flexible, but it does not break apart. A phospholipid is a type of lipid. It was mentioned in Unit 2, and it was mentioned there that we would discuss it further in Unit 3. A phospholipid has two fatty acid tails, plus a glycerol molecule, plus a phosphate-containing head, thus the name phospholipid. The phosphate-containing heads are hydrophilic, meaning that they are attracted to and interact with water molecules, while the fatty acid tails are hydrophobic, meaning that they repel water. This dual property molecule is uniquely suited for its function as forming the structure of membranes. We also refer to the membrane structure as being fluid mosaic. What this means is that under a high-power microscope, the membrane looks like mosaic tile artwork, which is tile set in mortar. Mortar is what holds bricks together in a brick building. If you have tiles on the wall of your shower being held by mortar, you might actually have mosaic tile artwork in your bathroom. It's said that the phospholipids form the mortar in which mosaic tile proteins are embedded. A phospholipid is a molecule with two fatty acids and a modified phosphate group attached to a glycerol backbone. The phosphate may be modified by the addition of charged or polar chemical groups at the position labeled R at the top. The internal environment of cells contains over 50% water, and most cells reside in watery conditions. Due to these watery conditions, a phospholipid bilayer forms with the hydrophilic heads oriented towards the outside of the bilayer, with the hydrophobic tails oriented towards the inside of the bilayer. Cholesterol, which is a steroid lipid, is also found in the membrane, and this makes the bilayer stronger yet more flexible. What this means is the bilayer is more fluid without coming apart. The phospholipid bilayer is the major component of all cellular membranes. The hydrophilic head groups, indicated by the small red circles, of the phospholipids face the aqueous, meaning water-containing solution, outside and inside the cell. The hydrophobic tails are sequestered in the middle of the bilayer to avoid making contact with the water outside and inside the cell. There are thousands of proteins either embedded within or attached to the surface of the phospholipid bilayer. Some proteins have carbohydrates attached. These are referred to as glycoproteins. Glyco as a prefix means sugar. Recall from Unit 2 that glycogen is the storage form of carbohydrates found in animals. The functions of these proteins include to help move substances across the membrane, either into or out of the cell, and also to communicate with other cells in the organism. There are 5 major types of proteins in the plasma membrane discussed here. First are transport proteins that move hydrophilic substances through the membrane that would be repelled by the hydrophobic fatty acid tails of the phospholipid bilayer. We discussed the details of these transport proteins in Unit 4. There are enzymes that control chemical reactions, as well as recognition proteins, which are markers that identify cells as self to the immune system. Consider the fact that your immune system has to be able to identify pathogens that make you sick as foreign and destroy them while leaving your own cells alone. Recognition proteins help your immune system distinguish between cells that belong in your body and those that do not. Adhesion proteins help cells stick together to form a cohesive layer in certain tissues, while receptor proteins bind to exterior signals such as hormones to deliver chemical messages to the interior of a cell. The eukaryotic plasma membrane is a phospholipid bilayer with proteins, glycoproteins, glycolipids, and cholesterol embedded in it. Take a look at the proteins listed here. A peripheral membrane protein is attached to one side of the phospholipid bilayer, while an integral membrane protein spans both sides of the phospholipid bilayer. On the right side, you see a protein channel which forms a tunnel through the phospholipid bilayer to allow hydrophilic substances to move into or out of the cell. Here we discuss eukaryotic organelles. These are membrane-enclosed structures in the cytoplasm that perform specialized functions. This is translated as little organs, so these are essentially the organs that allow your cells to function. The cytoplasm is a viscous fluid. It contains water, but the water is thick because it has many dissolved molecules and ions in it. The organelles reside within the matrix of the cytoplasm. We can also say that the cytoplasm contains all of the contents of the cell between the plasma membrane and the nucleus in eukaryotes. The nucleus contains the cell's DNA. We will discuss it more later. Different eukaryotic cells contain different organelles. For instance, animals versus plant cells have some differences in their organelles based on differences in cellular function. The figures shown on this slide and the next slide show the major organelles and other cell components of a typical animal cell and a typical plant cell. The animal cell shown here has lysosomes and centrosomes, which the plant cell does not. This figure shows the major organelles and other cell components of a typical plant cell. Notice that the plant cell has a cell wall, chloroplast, plastids, and a central vacuole, structures that were not found in the animal cell on the previous slide. Here we start discussing the specific organelles found in eukaryotic cells. The endomembrane system is a series of organelles involved in processing of materials for the cell. Endo means internal, so this refers to an internal series of membranes. What the endomembrane system does is it breaks the cells into compartments to restrict chemical reactions to specific regions. The endomembrane system includes the nuclear envelope, which surrounds the nucleus and is connected to the endoplasmic reticulum membrane. It includes the endoplasmic reticulum itself, the Golgi apparatus, lysosomes, and vesicles. Vesicles are tiny membrane sacs that enclose and carry molecules from one part of the system to another, thus connecting the different components of the system. The nucleus houses the cell's chromosomes, which are long DNA structures containing genes that contain the instructions to make proteins. While we discuss the details of chromosomes and genes in Unit 7, for now you should realize that all of the DNA in your cells is associated with proteins. This DNA protein complex is referred to as chromatin. The proteins are there to coil up and condense the DNA so that it fits inside your cells. If we were to remove all of the DNA from one of your cells and stretch it out end to end, it would be in length around 6 feet. This is longer than most of you listening to this lecture, so this means that each one of your teeny tiny microscopic cells have 6 feet worth of DNA. In order for this DNA to fit, it must be wrapped around these proteins. The nucleus is surrounded by the nuclear envelope, which is a double membrane that encloses and isolates the nucleus from the rest of the cell. Within the envelope are nuclear pores. These are holes that control the entry and exit of molecules from the nucleus. Small molecules such as water and ions are able to flow freely through the pores, while the passage of larger molecules such as proteins and RNA is controlled by proteins that reside in the pores. Within the nucleus is a specialized region called the nucleolus, where the components of ribosomes are manufactured. Ribosomes are the site of protein synthesis, and they are composed of proteins and ribosomal RNA, referred to as rRNA. Ribosomes consist of small and large subunits that once they are manufactured within the nucleolus, they exit the nucleus and are assembled in the cytoplasm to make proteins. They either become attached to the rough endoplasmic reticulum, which is discussed in the next section, or they stay free within the cytoplasm. The nucleus stores chromatin, which is the DNA plus proteins, in a gel-like substance called the nucleoplasm. The nucleolus is a condensed region of chromatin where ribosome synthesis occurs. The boundary of the nucleus is called the nuclear envelope. It consists of two phospholipid bilayers, an outer membrane and an inner membrane. The nuclear membrane is continuous with the endoplasmic reticulum. Nuclear pores allow substances to enter and exit the nucleus. The endoplasmic reticulum forms interconnected, membrane-enclosed channels in the cytoplasm. There are two types of ER, rough and smooth. Rough ER is called rough because it is studded with ribosomes that manufacture proteins. Once the proteins are made, they are packaged in vesicles that carry the proteins to different cellular destinations. The smooth ER is the site of lipid synthesis, including phospholipids and cholesterol. Lipids manufactured in the smooth ER are also packaged in vesicles and travel to the plasma membrane, the nuclear envelope, or other organelle membranes to replace worn-out areas within the membranes. As mentioned, the nuclear membrane is continuous with the endoplasmic reticulum. Notice the red dots. Those are the ribosomes on the rough ER. The rough ER is also connected to the smooth ER, which is not shown here. However, the smooth ER looks exactly like the rough ER, except it does not have ribosomes. That's why it's called the smooth ER. The Golgi apparatus consists of a stack of flattened membrane sacs. Vesicles containing the proteins and lipids that were made in the rough and smooth ER travel to the Golgi and empty their contents into the Golgi for processing as follows. The Golgi manufactures some sugars and attaches sugars to the proteins and lipids to yield glycoproteins and glycolipids respectively. Proteins also get folded into their final three-dimensional shape in the Golgi. The final molecules get packaged in new vesicles and are sorted to be sent to their final cellular destinations. The molecules can either stay within the cell or they get secreted outside the cell. An example of a protein that would get secreted outside of cells would be hormones. Proteins are synthesized in the rough endoplasmic reticulum. Vesicles bud from the ER and fuse with the cis face of the Golgi apparatus, which is the face closest to the ER. As the protein passes through the Golgi cisternae, which is the inside chamber, it is modified by the addition of carbohydrates and folded into its three-dimensional shape. After modification is complete, it exits via vesicles that bud from the Golgi's trans face, furthest away from the ER, and in this case the vesicle fuses with the cell membrane for secretion of the protein outside the cell. Since enzymes are proteins, they are produced in the ER. Specialized digestive enzyme proteins that break down different components are produced in the ER, travel to the Golgi, and in the Golgi are packaged into vesicles called lysosomes. The enzymes within the lysosomes degrade nutrients, excess membranes, defective or worn-out organelles, any other cellular debris, and even bacteria or viral components. Specialized white blood cells are loaded with lysosomes that break down these pathogens to clear them from your body. Here we see a cell ingesting a large food particle into a food vacuole via a process called phagocytosis. The food vacuole then fuses with a lysosome within the cell to break down the large food particle. While the cell does contain organelles, these are not shown here for simplicity. Vacuoles are large vesicles found in both animal and plant cells that contain different components such as water, food, or waste products. Central vacuoles are found in plant cells only. These are large vacuoles filled mostly with water to regulate the amount of water in the cell. For instance, after plentiful rainfall, the cell will store the excess water in its central vacuole to use during times of drought. The central vacuole also isolates hazardous waste products as well as stores poisonous chemicals which deter animals from eating the leaves. Consider poison ivy or poison oak, which contains some of these chemicals. Once an animal gets a rash on its mouth after eating one of these poisonous plants, it will no longer eat the plant. Lastly, the central vacuole stores excess nutrients such as sugars and amino acids. Peroxisomes are another specialized type of vesicle that dispose of toxic substances in all eukaryotes. One example is peroxisomes found in the liver and kidneys of animals that detoxify drugs, alcohol, and other poisons. Chloroplasts are the site of photosynthesis in plant cells. They capture solar energy from the sun and convert it to the chemical energy of sugar molecules. Chloroplasts are surrounded by a double membrane and are filled with a thick fluid called stroma, in which stacks of hollow membranous sacs called thylakoids reside. Chloroplasts also contain the green pigment chlorophyll that gives leaves their green appearance. The chloroplast has an outer membrane and an inner membrane filled with a fluid called the stroma. Stacks of membrane structures called thylakoids nestle in the stroma. Each stack is called a granum. All eukaryotic cells have mitochondria, which contain two membranes, a smooth outer and folded inner membrane. Mitochondria provide cellular energy by converting glucose to ATP or adenosine triphosphate. ATP is a nucleotide, but it does not get added to nucleic acids as we discussed in Unit 2. ATP is chemical energy in the form that is usable by cells. Hydrolysis, or breaking down of ATP, releases great amounts of energy. Here we see a highly magnified picture of a single mitochondrion as viewed with a transmission electron microscope. This organelle has an outer membrane and an inner membrane. The inner membrane contains folds called cristae, which increase its surface area. The space between the two membranes is called the intermembrane space, and the space inside the inner membrane is called the mitochondrial matrix. ATP synthesis takes place on the inner membrane. The last specialized structure to discuss in eukaryotes is referred to as the cytoskeleton. Cyto means cell, so this is a cellular skeleton. The skeleton in your body is made of bone and gives your body shape. Just like the cellular skeleton gives cell shape, however, it is made of protein fibers. The cytoskeleton also organizes the cytoplasm by attaching many organelles to the cytoskeleton within the cytoplasm. The cytoskeleton is made of different protein types with different diameters. Microtubules are the largest, intermediate filaments have an intermediate cylinder diameter, while microfilaments are the smallest. Functions of the cytoskeleton include cell shape, especially to determine and maintain cell shape in cells that don't have a cell wall and only have a flexible plasma membrane as their outer boundary. The cytoskeleton also aids in cell movement, such as swimming sperm cells and cells that need to circulate through the body. It also moves organelles, such as guiding vesicles, from the ER and Golgi to their final cellular destinations. Lastly, microtubules in particular move chromosomes during cell division. Here is an example of cytoskeleton proteins. These structures are called centrioles, which are cylinders made up of 9 triplets of microtubules. The non-tubulin proteins, indicated by the green lines, hold the microtubule triplets together. These microtubules are involved in moving chromosomes during cell division. Flagella and cilia are microtubule extensions that extend from the plasma membrane outside the cell. A flagellum is a tail that extends from the plasma membrane and spins to propel a cell for movement. Cells typically have anywhere from 1 to 3 flagella. Examples of cells with flagella include sperm cells in male animals that contain 1 flagellum, and bacteria cells that contain anywhere from 1 to 3 flagella. Cilia are also microtubule extensions of the plasma membrane. However, when they are present, they are much more numerous than flagella. Thousands of cilia line cellular surfaces to move fluids and particles over the surface using a wave-like motion. For example, in humans, cilia line respiratory tract cells going from our nostrils to our lungs to move mucus carrying debris and bacteria away from our lungs so that we can cough it out of our body or blow it out of our nose.